This is part 2 of Chapter 6, Lecture on the Cell, and we're going to take a look at a couple animations first. Okay, now we'll take a look at an animal cell. You can see actually by looking at this animation at the different structures inside the cell. So it's not really an animation so much as a way to explore a 3D model of a cell, sort of. So you can take a look at, oh, it will actually say what it is. So you can, interesting. Okay, so you can play with this just like I am right now. This is Learn Genetics. I'll show you the website right there. LearnGeneticsUtah.edu and Inside a Cell. It's actually a really good website to bookmark as a good resource because it does graphically lay things out for you. So we're going to now switch back to our lecture. After that brief little intermission, let's take a look at why we have organelles, and particularly in the complex eukaryotic cells. So they are specialized structures with specialized functions. For example, cilia or flagella have the function to provide locomotion. They act as containers. They can partition the cell into different compartments that are doing different jobs. They also can create a different chemical environment. So they can have a different pH from the rest of the cell, or they can concentrate materials or nutrients that are needed for a particular function in the cell. A lot of times, different functions that are occurring within a cell are incompatible with each other. And a good example of that is a lysosome, which is a vesicle that contains digestive enzymes. And so if those digestive enzymes were loose in the cell, they would destroy the cell. They would eat it up, digest it. So instead, they're concentrated within an organelle called a lysosome. And then the last thing is that membranes, again, act as sites for chemical reactions. So not only on the outside of the cell do we have that cellular membrane with all its properties that you've been working on, but also each organelle also has a double bilayer membrane. And so that membrane has similar properties to the outer membrane of the cell in that it can be a place where there are proteins and lipids located that are involved in different chemical processes. And so that organelle can then do its work based on whatever signal it's receiving or nutrients that it's receiving or whatever chemical reaction actually is going to help it get started in its job. And a good example of this is the organelles of chloroplasts or mitochondria that actually have their enzymes, a lot of them, embedded in the membrane that surrounds the chloroplast or surrounds the mitochondria. And some of the reactions that we have with photosynthesis or in cellular respiration are actually occurring in the membrane of that organelle. So it's a very important thing. It's not just a separation. It actually is involved in the function. Okay, let's see. I think that's all we want to cover there. So now basically some of the functions that a cell can take on are either to make proteins, to make energy, or to make more cells. So you can really make it that basic. You can divide up the different function of a cell based on these types of ideas. Of course, there is a lot more. For example, making more cells. You also have things like the lysosome that are involved in cleanup. So that is repair and renewal. It's not necessarily making a new cell, it's actually maintaining the one that's there. And so some of the functions of organelles is actually maintenance. I would actually add that to this list. So looking at building proteins first. Um, in order to build proteins, a cell has to read the instructions that are located in the DNA. That's done through translation. The RNA is translated into proteins within the cytoplasm of the cell. And then those proteins aren't finished yet. They must be processed. They must be folded, as we learned in our last unit. They will be modified. Sometimes it's taking some amino acids off. Other times it's adding things like carbohydrates to make a glycoprotein for the cell membrane. And also, it's important for proteins to act as transport proteins. And that's a big part, again, of what's occurring within the cell membrane as you guys have been building your models. There's a lot of transport across the cell membrane that's done specifically by proteins. So it's a very important part of a cell's function to correctly build its proteins. The organelles that are involved in this are the nucleus, where we have our DNA, the ribosomes, where the proteins are actually being translated. The endoplasmic reticulum is one place where they will be modified, oftentimes folded. The Golgi apparatus is another location where they are modified. And then the vesicles transport them from one organelle to another, and often out of the cell as well. So they'll take them for secretion. So these are the 1, 2, 3, 4, 5 organelles that are involved in protein building. This is another picture showing the different things that are involved. This is just focusing primarily on the nucleus. And so the structure of the nucleus is, of course, where we're going to find our chromosomes. We actually find them when they're not being translated. They're in a condensed form that we call chromatin when they're not being transcribed and translated. We have the nucleolus, which is where our ribosomes, our ribosomal RNA is actually being made. We have the nuclear pores, which allow the RNA to pass through, and also for nutrients to pass into the nucleus. Again, this shouldn't be moving. Every organelle like that really moved. Every organelle like the nucleus is going to have a double membrane, and so we have that with the nucleus. This is a close-up showing the nuclear pore. This is actually another picture showing those pores. And then attached to our nucleus, we have our rough endoplasmic reticulum. This is just going into the process of building that protein. So again, in the nucleus, your DNA is organized into its discrete units that we call chromosomes. And then each chromosome is composed of a single DNA molecule that is associated with specific proteins. Together, those proteins and DNA molecules combine to form what we call chromatin, and that chromatin is condensed into a very dense form that we call a chromosome. The nucleolus is located within the nucleus again and is primarily responsible for ribosomal or rRNA synthesis. Okay, and this is a similar picture. So we'll stop there. And this is just showing that process of protein building. So in a very simplistic model here, we have the nucleus, and then we have the ER, endoplasmic reticulum. So our protein process is going from nucleus to ribosome to processing in the ER to processing in the Golgi, and then excretion out of the cell. And of course, that's a very specific type of protein because not all proteins will be secreted out of the cell. Many are going to be used within the cell itself. This is a close-up showing the nuclear pores. Okay, now this is a little bit of a focus on the nucleolus. So it's very important, again, in terms of protein synthesis, because without ribosomes, we're not going to be able to make proteins. So the ribosomal subunits, as you remember, ribosomes made up of two, a large and a small subunit, that is consisting of ribosomal RNA and proteins. So those subunits together are what actually translate the messenger RNA into a protein. But they must be made in the nucleolus first and then exported out through the nuclear pore to the cytoplasm in order to make a functional ribosome. And there are two types of ribosomes. We have the bound, which are attached to the endoplasmic reticulum, and we have the free, which are located in the cytoplasm. This is just showing you that. Okay, and again, these are just things. You can stop and examine these pictures if you like, but this is showing you what we've been talking about. And this is reiterating the free versus bound ribosomes and their primary job. One's going to synthesize proteins that function in the cytoplasm or cytosol, and the bound is going to primarily make ribosomes that are exported. Also, when we get into ER, we'll also look at another function of the endoplasmic reticulum, which is to make the proteins that are bound for the cell membrane. So those types of proteins are also going to be made by the bound ribosomes on the rough endoplasmic reticulum. Okay, so a concept that perhaps you haven't heard about in previous biology classes is the endomembrane system. So the endomembrane system is very involved in regulating protein traffic, and it's also involved in the metabolic functioning of a cell. So essentially, it's what a cell is about is going to be controlled by this endomembrane system. So what it consists of are the 6 things that you see listed here. We have a nuclear envelope, our ER, the Golgi apparatus, the lysosomes, the vacuoles, and the plasma membrane. So together, all of those 6 make up the endomembrane system. And all of these are either continuous or they're connected by transfer of elements by vesicle. So what that means is that either these things are touching each other within the cell, or they are connected by way of vesicles that act as taxis between them. So taxis will go from vesicles, not taxis, will go from, for example, the endoplasmic reticulum to the Golgi, or vesicles will go from the Golgi out to the plasma membrane. So in that way, we consider this a continuous system. So now we're going to look at the endoplasmic reticulum. So, and actually we're almost done with this one, so we'll start up with the next lecture on that. So I'll end with, actually I'm going to just end.